Hello, everybody, and welcome back to Academic Biology with Mr. Adolph. We are going to continue with our cell division section, and we're going to finish up here with the last concept, and that is meiosis. Now, we spent the last part of this section talking about a process called mitosis and the cell cycle, and that is when you have cells that are going to split, and you're going to get one cell becoming two new genetically identical cells. Now, that works great for what we have called somatic cells, which are our body cells. But not all cells are going to divide that way. Some cells need to go through the special process, which we're going to call meiosis. So let's get started. In organisms, there are two types of cells, as we talked about in the last section. We have gametes, which are going to be our sex cells, which is what we're talking about now. And then we have our somatic cells, which are our body cells, and they go through mitosis. Now, not all organisms reproduce the same way we do. We reproduce in a process called sexual reproduction. And in sexual reproduction, you need two individuals to make a new individual. Asexual reproduction, one individual becomes two individuals. So one organism is going to split into two. This only works in single-celled organisms. So you're going to see this in little prokaryotes. So you're going to see this in bacteria. That's why bacteria is so good at reproducing. It can just double in numbers so quickly. Now, we're going to talk a little bit more about this farther into this lecture. That works great for some scenarios, but not all scenarios. We have evolved this thing called sexual reproduction. And in sexual reproduction, we need a couple things to be different. And that's why we need this process called meiosis. So in organisms that reproduce sexually, we have these things called gametes. There are specialized sex cells. In males, it's a sperm cell, and in females, it's an egg or an ovum. Now, because these sex cells have to produce a new organism, you can't have the same number as you have in a somatic cell. So in somatic cells, in humans, we have 46 chromosomes, and you get 23 pairs from your mom, 23 pairs from your dad, giving you 46. In your gametes, however, you can't have 46 because if you donated 46 and your husband or wife donated 46 to your new baby's going to have 92. And as we talked about before, that is not good. So in order to make this whole thing work, nature had to come up with a way to get that number down. So meiosis is what we have, right? And in meiosis, you end up with half the number of chromosomes that you do in a somatic cell. So in humans, that's going to be 23. We need to go through some terminology. And the first little bit here we're going to talk about is something called a haploid versus a diploid cell. So I'm just going to move this over a little bit so we can see that diagram up in the corner. Okay, so ploidy refers to number of chromosomes. So you might see ploidy written on something. That just means how many chromosomes are there. Di means two or double. Ha refers to half. So a diploid cell is a cell which has double set of chromosomes. It's got double the amount of chromosomes as a haploid cell. So these are going to be your somatic cells. Haploid cells means half, so this is going to be just one set of chromosomes, and this is going to be your gametes. So if you look at this diagram up here, you can see each one of these diagrams has 3 chromosomes in it. The difference is, in a haploid cell, it's just one copy of that chromosome. It's a single chromatid. And in a diploid cell, we're going to have what we call 2 or sister chromatids. So the difference between these two is they both have 3 chromosomes. The difference is in the haploid cell, it has a single copy of each chromosome, or chromatid, I should say. And in a diploid cell, it has a double copy of each chromatid, giving you a sister chromatid. Now, if I were to take 2 chromosomes that are the same as each other, those are going to be called homologous chromosomes. So if I have 2 diploid chromosomes that are the same as each other, that is going to be our homologous chromosome. Homo means same, hetero means different. So homologous in this case means that 2 chromosomes that have the same genes. So we're going to talk more about that as we go through. Let's talk a little bit more about meiosis here. Okay, so what is it? So meiosis is very simply a process that has been evolved in order to make gametes or haploid cells. In mitosis, you have a cell which divides into 2 genetically identical daughter cells. So you start with one cell that has 46 chromosomes, and you end up with 2 new cells that have 46 chromosomes. In meiosis, that is not the case. In meiosis, you have a single cell, which after the whole process, you end up with 4 new haploid daughter cells, each one with half the original number of chromosomes. So the first cell had 46. The 4 new cells that are formed from this process are going to have 23. And the way this occurs is by going through two sets of divisions. So in mitosis, a cell divides one time. One cell splits into two. In meiosis, one cell splits into two, and then it splits into two again, and you end up with 4 from 1. So a diploid cell goes through two divisions. So if you look down here, you can see that, all right? So this will be the original cell here, which is an interphase. It doubles its number, and you end up with your sister chromatids here. So this would now be a diploid cell, right? 2N, and it's going to go through a process called mitosis. It's going to, this is phase 1 of meiosis. It's going to go through the first phase of meiosis, and what you're going to end up with is 2 new cells. Now, these new cells are a little bit different than in mitosis, because in mitosis, you end up with 2 new cells that are going to be diploid. In this case, we have cells that are diploid, but they're going to have half the number of chromosomes. So for what I mean by that is these are both going to have 46 chromosomes, but only 23 of the actual chromosomes. It's going to have 46 chromatids, but only 23 X's. And those 23 X's are now going to go through a second division. Now, here's what's different. It doesn't go through interphase, so that DNA doesn't double. So instead of going from 92, 46 to 92 again, it goes from 92 to 46, and then it immediately immediately cuts in half into another set of divisions, which is meiosis 2, and you're going to end up with 4 new cells. Each one of these new cells is only going to have 23 chromosomes in it. So from 46 to 23, you get 4 new cells. Let's talk a little bit more about meiosis. Now, during meiosis, something happens that doesn't happen in mitosis. These sister chromatids are going to find their homologous pair. So, for example, this would be the father's chromatid here, sister chromatids. This would be the mother's sister chromatids, and they're going to be for the same genes. So this would be, let's say, chromosome 22, or it doesn't matter which one, or chromosome 18, and they're going to find each other, and they're going to pair up into this thing called a tetrad. Now, why is it called a tetrad? Well, if you've ever played Tetris before, which if you haven't, you're blowing it because it's awesome. But Tetris, in order to get a Tetris, you have to get 4 lines. Tetra means 4. So a tetrad has 4 chromatids, 2 from the father, 2 from the mother. And they pair up like this, and they form this little tiny structure called a tetrad. Now, why is a tetrad so important? Well, the reason why it's important is because what you're seeing happening in the second part of this diagram. When these two chromosomes pair up with each other and form a tetrad, they're going to come into contact with each other. And when they do that, something happens called crossing over. We talked about this before when we talked about chromosomal mutations. It's called a translocation, where part of the chromosome from the mother and part of the chromosome from the father are going to switch places. So when they separate from each other, the father's DNA here is going to have a little bit of the mother's attached to it, and the mother's is going to have a little bit of the father's. Why is this important? Well, what this allows for is that very important thing called genetic variation. It's like shuffling a deck of cards. If you've ever played poker before or any card game, you know in between rounds you have to shuffle up the deck because you don't want to get the same cards again. Well, when you make a new human or a new organism through sexual reproduction, we don't want them to be the same as the original. So in asexual reproduction, when a bacteria cell replicates, you get 2 new identical bacteria cells. When a human replicates, you get a new organism, a new person that is different from the parents. That is because of crossing over. Think about your siblings. If you have siblings that are from the same parents as you, they don't look the same as you. They don't act the same as you. That's because of this process. Think about what's happening right now with coronavirus, right? We're going through a very serious pandemic right now. And think about how some people seem to be fine if they get it, right? They don't even get affected, and some people are dying. The difference is immune systems. So some people have strong immune systems for some instances, some have weaker immune systems for some instances. And what that allows for is survival. If everybody had the exact same immune system and it killed one of us, well, guess what? It's going to kill all of us. And that is the advantage to sexual reproduction. It allows for organisms to have variation. Let's talk a little bit more about this. So how does this whole process work? Well, it's 2 separate divisions. So during the first division, which we call meiosis 1, you get a diploid cell which has 46 chromosomes or 46 X's. It's going to go through a division just like mitosis, and what you're going to end up with is 2 new cells. each one only has 23 X's. So it goes from 46 X's to 23 X's. Now, it still has 46 chromatids, right? So it's still going to, have that number, but instead of it being 46 individual chromosomes, they stay as X's. And then they're going to go through a second round of division, and that's the important one here. It does not go through interphase before it divides again. So instead of doubling that DNA, so it does not have a chance to double its DNA. So now those 23 X's are going to get divided up, and you're going to end up with 4 new cells, and each of those new cells is only going to have a single chromosome. So you're going to have 23 single chromosomes. So in that first division, you're going to end up with 46 becoming 23 X's, but those X's are still going to have that same DNA in there. And then you're going to end up splitting it again without doubling the DNA. And what you end up with is haploid, which means that there are 23 single chromosomes in those new cells. And they're genetically different from the original cell. So that is meiosis. On the next little set of lecture I'm about to give you, we're going to finish this up by comparing it to mitosis. And we're going to talk about karyotypes. So I will see you guys momentarily.